Good morning, everyone. Good morning, everyone. I'm Arne Kumigai, Vice Chair of Education in the Department of Medicine, and I have the absolute privilege of opening a special grad rounds, the FM Hill annual lecture for the Department of Medicine. I will shortly be turning this over to Dr. Paula Harvey for the introduction of the speaker. But before I do, I would like to spend a few moments with a land acknowledgement. The Department of Medicine recognizes that many Indigenous nations have long-standing relationships with the territories upon which the work of the University of Toronto Department of Medicine is conducted that precede the establishment of the U of T. We acknowledge our presence on the traditional territory of many Indigenous nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples. Today, this land is home to many diverse First Nations, Inuit, and Métis peoples, and we are grateful to have the opportunity to live, work, and gather on these territories. We acknowledge that Toronto is covered by Treaty 13, signed by the Mississaugas of the Credit, and the Williams Treaty, signed by multiple Mississaugas and Chippewa bands. Land acknowledgements are only a starting point for larger conversations. More concrete acts of restitution transformation are needed to address underlying inequities and blatant discrimination in the distribution of resources between Canada's First Peoples and settlers. At this point, I would like to turn things over to Dr. Paula Harvey, Chair of Medicine. Thank you.
Thanks, Arno. I'd like to welcome everybody this afternoon to Citywide Rounds. This year, as in the past, we've asked our Women's College Hospital, Dr. F. Margaret Hill, annual invited lecturer, to give this citywide talk today. The FM Hill Lectureship is held annually to honour an exceptional physician, mentor and teacher, and the former PIC at Women's College Hospital, affectionately known as Dr. Peggy Hill, who passed away in 2012 at the age of 93. For 27 years, Dr. Hill practiced at Women's as a teacher, clinician and researcher. And when she passed away, an endowment was established in her name. This fund has established 3 chairs that include the FM Hill Chair in Academic Women's Medicine, the FM Hill Chair in Health System Solutions, and the FM Hill Chair in Humanism Education, along with this lectureship in medicine and 2 resident awards. This year, Dr. Varuna Parekh, who I believe is joining us today, who was our previous wonderful Chief Medical Resident, was the recipient of the FM Hill Humanitarian Award, and Dr. Tosin Oceantoken, who also was attending, and Dr. Asfanjay Mufti, who I believe sends their regrets, were the recipients of the FM Hill Mentoring Awards. This year, we've invited Dr. Rulan Parekh to be our lecturer, and it is really my great honour to introduce Dr. Parekh. Dr. Parekh was appointed Vice President Academics at Women's College Hospital in September of this year. She's a professor in the Departments of Medicine, Paediatrics and HPME. She's a clinician scientist and an international leader in clinical epidemiology and translational research in kidney disease. At the University of Michigan, Dr. Parekh trained in pediatrics and internal medicine with subspecialty training in both pediatric and adult nephrology. She also completed her master's in biostatistics and study design. She subsequently went on to postdoctoral training at Johns Hopkins and joined the faculty there in 2000. She was awarded the NIH Career Development Research Award and the American Society of Nephrology Karl Goschalk Award for Young Investigators, and went on to establish a very successful research program in genetics and cardiovascular complications of kidney disease. In 2008, we were lucky she returned to her native Canada and accepted a faculty position at the University of Toronto and a scientist position at SickKids Research Institute. She has maintained a stellar record of sustained NIH and CIHR funding since 2000, and she's been the recipient of awards for research, teaching and mentoring, and was elected into academic honor societies such as the American Society of Clinical Investigation, the Association of American Physicians, the Pediatric Academic Society and the American Pediatric Society. She previously held a Canada Research Chair in Chronic Kidney Disease Epidemiology at SickKids, and she now holds the FM Hill Chair in Health System Solutions at Women's College Hospital. It is really with great pleasure that I introduce Dr. Parekh as this year's FM Hill lecturer. And today she's going to talk to us on race, ancestry, and kidney disease, genetic epidemiology to clinical translation. So thank you, Rulan, and I'll hand over to you.
Thank you so much for that kind invitation to speak today, and also to have the chair in Health System Solutions. I'll talk about Dr. Hill at the end of the lecture. I have no commercial interest to speak of. And I want to sort of give some background in that in nephrology, we've had this reckoning this past year of the use of race in one of our kidney disease based estimated GFR equations to estimate what kidney functions like. And what we've really grappled with over the years is this issue of racism within nephrology, how we're taking care of our patients, and how we really need to redirect how we're doing things. When you think about race-based medicine, and really this is from a Lancet paper in 2020, talking about using race in epidemiological studies that link race with disease, or biological studies that link race with biology, it's really this concern that, in fact, we're conflating issues of social determinants of health and other factors that cluster in certain groups, but we're not addressing the specific racial, the structural racial issues that are actually causing these issues. And in fact, we're attributing a biological mechanism to something that actually could be completely structural racism in the way that people are being treated. And so what's important to recognize is that now we're really thinking about this race-conscious medicine, in that race is the social and power construct, and how does that actually impact patients directly? I wanted to address this head on, not only because I, as the FM Hill lecturer, but also as the VP for Academics at Women's College, we are really motivated, and I came to Women's really to address inequities, and that I was interested in seeing how we could improve that overall for both our patients, as well as for our trainees, as well as for the faculty. What's important with the nephrology, and we're working with Born August and Jeff Pearl with the Canadian Society of Nephrology to really address what should be the race-based equations that we use to estimate kidney function, but in fact remove race completely and make them race independent. And we've been working on that for the last few months and hope to have something come out soon in order to show what we think we should do in Canada. But what has been really clear is that there's been an inappropriate use of race within nephrology, and that this colleague of mine at UPenn, Amaka Anenye, has really talked about how structural racism and racial discrimination has led to some biological impact. In fact, with the constant exposure of microaggression, that you're able to get increased sympathetic nervous activity, altered metabolism of insulin and other hormones. There's also social determinants of health, such as food insecurity, access to healthcare, which is an enormous issue in the United States. And that ultimately has impact to this altered kidney pathophysiology. But it's important to recognize also that among individuals, that it isn't consistent, and that race alone does not capture all of this information. There was also an article in 2021 in the New England Journal that said, absolutely that race, ethnicity are incredibly important, and we have to think about structural racism. But when you think about genetics or ancestry, that there is this complex intertwined relationship, and it demands this sort of nuanced analysis, and it's complicated. The other thing that happens is, as we talk about precision medicine for our patients, if we don't understand genetics or ancestry, what happens is that we may leave some people out, which leads to further inequity. And so we really need to think about, when is it appropriate to think about genetic ancestry, and when should we be addressing these issues? And my hope is to tell you how I've been addressing it over the last 20 years. And what I hope is moving the field forward, but trying to do it carefully. And areas that I'm concerned that we may be conflating issues, and that I really want to raise that as a red flag to the nephrology group, that we really need to think about it differently. So I'm trying to use an informed use of race, ethnicity, and ancestry to recognize that there are terms of race that gets used. I'll use that when they're historically used and self-ascribed to populations I've studied in the US, when people have reported as African Americans or West Africans or from specific countries. And primarily, I'm going to try and focus as much as I can on gene variants and really try to describe the gene variants that are associated in certain groups, and how we've been studying that to be able to understand genetic risk of disease. These are great references. Most of them have come out recently. And I think it's worth reading as we really think about now this race conscious medicine and how do we actually address it with our patients. So let me tell you about nephrology. I think it's important to know that the burden of disease is enormous. There are about 850 million people in the world that have chronic kidney disease. We do not have enough nephrologists, staff, trainees, nurses to be able to deal with all of those millions of people with kidney disease. We're fighting hard with the International Society of Nephrology, with the WHO, to really make kidney disease a non-communicable disease, because we really think it's important that, in fact, it's addressed worldwide. And given the burden of disease in many countries, especially those with low-income countries, it's really important for us to address kidney disease as a significant impact on overall mortality. It actually leads to an increased risk of not only overall mortality itself, but it also increased risk of cardiovascular disease. And so it's really an important public health issue that we're able to address this. Specifically, when you look at risk of mortality among millions of persons with chronic kidney disease, if you take a look at the left figure, you can see that when your kidney function, which is an estimate of your GFR, your glomerular filtration rate, when it's about 120 to about 75, that your risk is basically only a little bit higher if your protein in red is a little bit higher compared to normal protein. But you can see the moment your kidney function starts going less than 60 mils per minute per 1.73 meters squared, that there's this rapid inflection and the risk goes up as high as 4 to 8 times more likely risk of death. And even so, on the right, for cardiovascular mortality, there's a significant inflection. And it's really important to note that the risk of mortality is incredibly high from cardiovascular disease. And many of these patients actually die of cardiovascular disease before they even get to end-stage renal disease, where they could be treated by dialysis. When I first practiced actually as a faculty member, I was in Baltimore, which actually had one of the highest rates of disease within the country. In fact, I lived in the city of Baltimore and 100% of my patients were Black. They were African-American and they were from the city of Baltimore. And it was remarkable to me that it was so common, and many of them actually talked about a family history of kidney disease among themselves. What was obvious at that point from my clinical practice, but actually taking a look at it now within the United States, you could see that in fact the prevalence of end-stage renal disease was quite high, but it was about 4.2 times greater among those who are African American than compared to whites in 2006. And it has improved slightly over the years, which is great, but it's a slow progress in addressing this disparity in end-stage renal disease. Native Americans, which are here in gray, have a 1.3 times greater prevalence than whites, and Asians also have, in green, have a 1.3 times greater prevalence than whites. So it's important to note that there are disparities still within end-stage renal disease, but the greatest disparity are among those who are African American. There was also a study called the African American Study of Kidney Disease that was conducted in the late 1990s. And in this, they were actually studying African Americans who had some mild chronic kidney disease, and they were able to put them onto a clinical trial of ACE inhibitors and a calcium channel blocker, give standard controlled blood pressure or intensive controlled blood pressure. And in fact, during the trial phase, here you could see that those patients who were on intensive trial and were receiving an ACE inhibitor actually did much better than those who were on a calcium channel blocker. And in fact, the trial got stopped early at 3 years because there was such a significant benefit among those on an ACE inhibitor and had intensive therapy. What happened over time, though, was that as you followed people over time, you could actually see that, in fact, there was still this relentless progression that happened. And in fact, 50% of the patients who were in this trial ended up going on to end-stage renal disease. And so they followed patients up to a decade. The patients that are still alive are still being followed. But in fact, almost all of them have either had cardiovascular mortality before they reached end-stage renal disease or have reached end-stage renal disease. What we also know is that there's a specific disease called focal segmental glomerular sclerosis. This is a disease that presents with nephrotic syndrome where you get massive proteinuria. You are quite edematous when you present. And when you actually take a look at the kidney biopsy itself, you can see multiple variants that can occur. In fact, one is called the collapsing variant, which is seen among those persons who are infected with HIV or COVID. And what's difficult about FSGS is managing it sometimes, and that there's a subset that rapidly progress to end-stage renal disease. There are also significant comorbidities due to the massive proteinuria. And in fact, so far to date, about 40 genes have been identified, mostly autosomal recessive, with the association of FSGS. In this figure on the left, you could actually see that the absolute numbers are much higher in those who are deemed white versus African American. However, if you were to take a look at the incidence rate, the rate is much higher, in fact, 3 times greater among those who are Black. And so this was a study that went up to 2000, and we suspect that these numbers actually continue to rise over time. And so again, for some reason, there was also this difference in incidence among patients who were African American compared to white, and it was unclear why there was that difference. So what we do know is that kidney disease is heritable, and without giving you all the studies to lead into that, is that we've seen clustering of kidney disease in families with nephrotic syndrome, FSGS, IgA, and nephropathy, and end-stage renal disease. And in fact, when you take a look at asking the question of family history of a first-degree relative, it actually increases your risk of end-stage renal disease by 2-fold, and but 5 times greater among those who are African American. So based on that, we really thought that kidney disease was heritable. There was something specific that we needed to take a look at, and we needed to understand why there was such a greater burden of end-stage renal disease. So why study genetics? I think what's really important to understand is that kidney disease carries a really high mortality. And for us, when patients reach end-stage renal disease, in fact, the risk of mortality in 2 years for those patients who have diabetes is 50%, which is incredibly high. And so we are always trying to understand the mechanisms and potentially think about druggable targets. Our goal is really to try and prevent those patients progressing on end-stage renal disease. We also do genetics because you can potentially screen and ultimately decide who should get therapy, or what therapies could you modify that you currently treat with, and maybe that's different. And then the question is it predictive of progressive disease and end-stage renal disease? So maybe you could use it for kidney transplant donors. So one of the things that I want to give an example of is this is a discovery of a gene, a rare variant that had a large phenotypic effect in a common disease. So Helen Hobson and John Cohen took a look at the Dallas Heart Study. They were taking a look at rare variants. On the left-hand side, they were able to take a look at, in about 3,300, the distribution of LDL cholesterol, which is known to be associated with cardiovascular disease. When they take a look at then the PCSK9 variant, they were able to show just in 85, this very rare variant, that was a significant reduction in your LDL cholesterol. When they looked at it further and said over 15 years, was there a difference in those who had this variant versus those who didn't? And in fact, there was a significant, highly significant difference in the prevalence of coronary artery disease. And what's important is that the biology of this actual region was identified and worked on by actually a number of scientists in Montreal. And based on that work is actually the development of all the medications now for the PCSK9 that have had a significant impact in treating coronary disease. And in fact, it's really among those that are getting that have this variant that you could see then, in fact, over age, that this is probably a variant that can definitely impact your long-term risk for cardiovascular disease. So this is what we would hope that we would find within kidney disease, but unfortunately our story is a little bit more complicated. On average, it's really important to recognize that we all share 100%, almost 100% of the genome. We all share exactly the same 25,000 genes among all of us. When we talk about genetic risk, we're really trying to understand what's the 1% variation that occurs when we look at different populations and their allele frequencies and these different alleles. But we recognize we are all exactly the same. In trying to understand this 1% difference, what we really hypothesized, this is back in the early 2000s, was that using admixture mapping, which is really a old school version of genetics before we were able to do everything high throughput now, we took a look at an admixed population and said, let's see if we can come up with this map of admixture and actually identify different regions, ancestral regions, and see if that's associated with kidney disease, with the hypothesis that African Americans were an admixed population. So what we looked at is 2 African chromosomes, 2 European chromosomes, and then ultimately over multiple generations, you would get admixture, and just here would be the variant that we would be interested in, and you could see that it would change over time in a multiple, sorry, going backwards, in a multiple, in multiple generations, you would see that you would be admixed, and ultimately your population would be blue and yellow, and that you could see then the marker was then inherited at certain periods. What we identified then was the chromosome 22 locus for end-stage renal disease, and it was important to know that we really engaged the community at that time, before you called it community engagement, and we really worked with the community to identify how would things be getting defined, what are the questions that we would be able to ask, making sure that we were able to really address something that they thought was important to them. And we recruited about 2,000 spousal controls and those patients with kidney disease on dialysis, and frequently heard about this clustering of disease. And so for them, this was quite important for us to have identified, predominantly within the Baltimore area. We then took a look at these genes specifically and then focused in on the ones that were known to be associated with Mendelian disease, which is called MYH9. And right beside MYH9 is a gene called APOL1. In fact, they're in tight linkage. And we didn't have all the 1,000 genome data at that point. And so when the 1,000 genome data came out, some colleagues in Boston actually were able to identify that perhaps it wasn't just mutations in MYH9, but it was mutations in APOL1 that were associated with kidney disease. And it gave a great story, because if you had variants in this APOL1 gene that were associated with chronic kidney disease, there were actually two SNPs, two polymorphisms within this G1 haplotype, a deletion polymorphism within this G2 haplotype. But what was important to recognize was that it protected you against sleeping sickness. And so if you didn't have any risk alleles, you could actually get sleeping sickness. If you had one of these kidney risk alleles, you didn't get sleeping sickness. And if you had two risk alleles, again, you didn't get sleeping sickness, but then you got chronic kidney disease. So similarly to malaria, we thought that there was potentially this advantage, and a lot of data had come out from the original publication to 2 years later to identify this region as potentially protective. It's important to understand that APOL1 is bound to HDL and really allows it to circulate. And then also it's present in all humans and also some non-human primates. So it really is a higher level order of a protein that's being developed. So is it analogous to sickle cell disease? I actually think it's quite different because sickle cell will give you an acute crisis. This is more of a progressive disease. They're both autosomal recessive. However, you get complete penetrance in sickle cell disease, and we have very variable penetrance in APOL1, which I'll go over. One variant rarely causes disease, even though that's questionable now with sickle cell trait. There's no risk at all with one variant. You're at risk for getting sickle cell and chronic kidney disease. But what's more striking is actually the prevalence of the variants are quite relatively rare among African Americans, whereas much more common among African Americans. And in European Americans, it's uncommon, but still present, and similarly for APOL1. So since that time, over the last 11, 12 years, there have been a huge number of papers, an explosion of papers, which has been great to see that everyone's really interested in this, because we're all really trying to understand the biology and understand who we should be measuring APOL1 on. So you'll be happy to know that I'm actually not going to review all 573 papers, but what I'm going to try to do is actually just give you some snippets of things that I think are really critically important and things that have come up in the last year or so that are really important for us to think about also here in Canada. So going back to why we want to understand the gene, it's really the biology, the disease, and the druggable targets. And what's interesting is over the last 10 years, there have been several mouse models expressing APOL1 G2 deletion that have been able to recapitulate proteinuria and kidney disease. So it's good. So we think it's actually causal. It's not just a marker of association, as we found in the genetic epidemiological studies, but we think it's causal. Interestingly, the G2 deletion leads to more disease than G1. And so there may be allelic differences, which have not been able to be tested in humans yet, because really there's not enough of those who've got G2. And similar to humans, though, it's also an incomplete penetrance in mice, as not all the mice are affected, which is important. And the other thing is that they're conditional variants, where you actually have to have a specific environmental stimuli in order to have a disease-inducing function. And so this is really important to understand, is that maybe an HIV disease where you have a large inflammatory component that actually stimulates the gene itself, it may stimulate how APOL1 functions, and therefore that leads to having having kidney disease or developing progressive kidney disease. And so it isn't just having the gene itself, but you actually need to have this second hit. What we do know about APOL1 is that currently, if you have it, again, it protects you against sleeping sickness. So specifically taking a look at rodenziense, which is the type of trypanosome, what happens if you have APOL1, the wild type, you actually enter into the trypanosome itself, you actually bind to the virulence factor, and nothing really happens to the parasite itself. But if you carry the variants that are important, then when you enter into the cell itself, you're not able to bind to the virulence factor, the lysosome of the parasite swells, and then the parasite dies itself. And so it's important to recognize that that's really the function of this, of the APOL1, and that it binds to the cell itself, enters into the cell through a pore, and then is able to bind to this factor. What's confusing is that this is really how APOL1 is protective against rodenziense, which occurs on the east coast of Africa, but it isn't that protective against gambiense, which occurs on the west coast. So that really flies in the face of our hypothesis of West Africans being at higher risk. So we still need to try and understand why there is this difference. What we do understand now, and this is just a quick summary of almost 10 years worth of work of Martin Parekh, Friedman, and Kathleen Sustak, and Opi Oyemi and Duke. APOL1 risk variants actually form a pore and cause cell lysis so that the APOL1 can actually enter into the cell. When it enters the mitochondria, it opens a pore in the inner mitochondrial membrane and may lead to worsening, may lead to mitochondrial dysfunction. It's not clear how. It also misfolds in the endoplasmic reticulum and causes distress within the ER itself. And it's also been shown to affect the cytoskeleton of the podocyte potentially and leads to podocyte dedifferentiation. What just has been published in the last few weeks was that, in fact, if you were exposed to COVID or bacteria, you also get a cytokine storm, and then it increases APOL1 that enters into the endothelial cell. It increases the inflammasome itself, and basically leads to increased inflammation, poor adhesion, increased permeability in the endothelial cell. And the question is, does this ultimately lead to end organ damage and severity of sepsis? And so we don't quite know really all the mechanisms. We do know this entrance into the cell itself. And so what we've learned, though, before that, is that we really need to understand some basic mechanisms in order to be able to understand how to treat it. And before we've actually gone further, there is a company that I'll show you in a second that's actually using some of this basic biology and has already tried to address if we can treat the disease. I'm showing COVID because what would not be without discussing COVID in the last 18 months, but it's important to recognize that COVID has actually shown that those have, there's been a COVID kidney phenomena where you get COVID infection and get significant proteinuria and acute kidney injury. When these patients have been biopsied, they in fact show collapsing glomerulopathy, very similar to an FSGS picture, very similar to HIV disease when we had very aggressive HIV disease. And what they've been able to find is that, in fact, all of these patients have been high risk of some sort, and perhaps APOL1 is associated with COVID itself. Not infection, but COVID kidney and a proteinuric disease. So going back to this company that actually has developed this protein Vertex is the pharmaceutical industry. It's actually the company that actually developed a lot of the biology that was required to understand how to treat cystic fibrosis. And what they've done is that they've been able to create this molecule called VX147 that binds to recombinant APOL1. They've been able to show as it binds to it that across the dosing of it, that they've been able to bind to it at all times. They've also been able to show in a mouse model that's been induced by interferon gamma, that they've been able to actually induce APOL1 expression. And as they induce APOL1 expression, you can see then your proteinuria actually goes up. I'm so sorry. Your proteinuria goes up over time. They've been able to show that potentially that your interferon gamma is actually really stimulating this massive proteinuria, which is important compared to the controls. Then what they did was that they gave the mice the VX147, and they were able to show that, in fact, this was your vehicle, but once you got the drug itself, then you were able to reduce the proteinuria quite significantly. And again, they were able to show the same reduction here within the proteinuria itself. So the hypothesis was that you take this healthy glomerulus, you take these two APOL1 risk variants, which enter into this ion flux mediated channel, pore, they enter into the cell itself and they cause this injury. So this is the first time we've actually had something within nephrology for APOL1 that we think potentially could actually lead to treatment. And unfortunately, the data is not out yet, but we've had there are 44 sites that are included. We were not one of them because we had a very small number that were here. Only 16 were enrolled, and it was very complicated to get patients. I'll show you why in a second. And the dose is given at dose 1 for 2 weeks, and then another one for the following 11 weeks. They are taking patients ages 18 and older. You have to have the APOL1 genotype, but remember, it's a very small proportion of the population, about 13%. So you've got to screen like 100 people in order to identify the ones that you need. You also have to have FSGS by kidney biopsy, and you have to be very early on within your course. And so the goal is to really look at this reduction in proteinuria of those 13 weeks. And the hope is that we're actually going to see some of that. And they think that that will be enough to be able to see enough reduction. And so the data will be out soon. Enrollment's complete, and now they're in follow-up phase. So we're hoping that this may be actually the first druggable target that we've ever had, targeting to be able to treat kidney disease. So it's exciting. And it's really we have very few trials that are positive in kidney disease. So this is something that we are quite excited about. So the next thing is, I'm going to show a lot of things about genetic screening to ultimately decide who do you provide therapy to or how do you modify current therapies. And the question is, who should be screened? When are they at risk? And under what conditions is there an increased risk for end-stage renal disease? So it's important to note again that you require a second hit if you've got APOL1 high risk 2 high risk alleles. That in fact, it's really important to note that it's key but not sufficient to lead to end-stage renal disease. It's a recessive pattern of inheritance. About 1 in 10 to 15 are high-risk carriers, and the incidence is about 830 per million persons who have kidney disease, advanced kidney disease. You have to have some environmental exposures, then ultimately you lead to chronic kidney disease. You can have this FSGS, albuminuria, or acute kidney injury. I've taken a lot of the studies that have been available and really trying to show where your risk is. Predominantly, these are odds ratios because they're mostly case control or cross-sectional studies. And it's been shown now that in preeclampsia, in children with hypertension, in young adults with FSGS and chronic kidney disease, with mild albuminuria or advanced chronic kidney disease, that in fact the risk is about 2 of having evidence of APOL1 association with kidney disease. However, when you start taking a look at populations that have HIV disease, perinatal HIV, or sickle cell disease, or even this collapsing variant, that in fact the risk is much, much higher. And that's really why the initial trial is based primarily on those who have FSGS, because we think that's really where the benefit is going to be when it comes to treating them. But it's also important that we're now able to also show that in fact APOL1 related diseases across the spectrum of age is also across the spectrum of different types of disease. And it also is primarily focused though on end-stage renal disease. One of the things that wasn't known though was how good is it at predicting sort of long-term outcomes. And what we were able to show was that if you had 2 copies of APOL1 risk variants, was that among African Americans in that initial cohort I showed you about the African American study of kidney disease, among that same population of about 700, you were able to see that if you had 2 copies over 10 years, you were at greatest risk of progressing to end-stage renal disease versus 0 copies or 1 copy. So you're about 2 times greater risk overall. In fact, if you look though at proteinuria, how many had no proteinuria or severe proteinuria? Proteinuria still was a very important risk factor, even more so than your APOL1 risk. And so it's important to recognize that that was something that wasn't interacting with your genetic risk, but it really showed how important proteinuria was to progressive kidney disease. In a most recent sort of meta-analysis, taking a look at a few of the studies we've worked on to say which are the ones, there are very few that have actually looked at progressive disease. The majority of those 573 papers that I highlighted are a lot of cross-sectional case control studies. And there's only a few that have looked at long-term outcomes. And it really looks like your long-term outcome of chronic kidney disease is about 1.4 times greater risk, of progression is about 1.7 times greater risk, and reaching end-stage renal disease, which is the most important, is probably about 1.65. However, it does cross 1. So overall, your risk of having advanced disease is about 1.6 times. And how fast do you decline? Well, I think this is probably most interesting for the nephrologist, but the amount that you decline is actually probably less than a ml per minute per 1.3 meters squared. So not that fast. And so we think it's a relatively slow decline over time. And in order for you to have APOL1 related disease, it's probably decades that we would be talking about. So one thing that's happened when we look at the effect size of APOL1 is that everyone was convinced that this was it. This was a single gene that sort of accounted for everything. But there's been regression to the mean, and I think it's really related to the study design. People's sample size have been relatively small, and as we get larger sample size, we're getting more precision. It really depends on the population, the difference between FSGS versus albuminuria. Depends on whether you start with some impacted kidney function as you start, and it also depends on your ancestry. And so the initial studies were predominantly case control, and some of them were quite large. Our initial one was in the thousands. But what's important is that there's some inflated odds ratios because it's such a variable disease, but it's also so variable because of all of these factors. But when we look at cohort studies, population or disease-based, we're able to say that the hazard ratio is 1.6, which I think is probably closer to the truth. And the annual decline is a relatively modest amount, but over decades will have a definite impact. So we're really interested in understanding who should be screened and when they should be screened and what else we should think about for the second hit. And so what's important is to understand this relationship between cardiovascular disease and kidney disease, in that both having APOL1 risk, environmental factors could lead to hypertension, and vice versa, hypertension and kidney disease are closely interrelated. And so the question is, if you take a look at APOL1, and you want to see the association with chronic kidney disease, the question is, do you get chronic kidney disease second and then get hypertension? And is this the second hit that you need, or do you actually get hypertension first and then get a second hit? And so what I'll look at is just the studies that we were just looking at to say, well, you already have kidney disease and then do you get hypertension? What's interesting is that there was no difference in those populations if we did standard therapy or intensive therapy. So perhaps maybe the degree of hypertension, how we managed it, really had no effect. Also, we took a look at the drugs that we prescribed people and said, did it make any difference in how we manage the hypertension by giving you ACE inhibitors or not ACE inhibitors? And we really didn't see any effect again. And then those patients were actually followed up to 5 years. And I just showed you the data up to 5 years, but now up to 15 years. The question was, did even tighter blood pressure control over time make a difference? And there might be some benefit in those who are high risk. They seem to do a little bit better if they had tighter control. So maybe there is a benefit. However, it's borderline, and I'm not sure that'll stand the test of time. So we're going to have to see about blood pressure after kidney disease, but maybe there's actually something related to hypertension before you get kidney disease. And so this was a study that was done in New York City and then compared to a population through a pharmacology consortium. And so they tested first among 5,000 African Americans and then tested again and validated among 3,000 African Americans. And they were able to show that in those who have younger ages versus older ages, that in fact that there was a relationship only in sort of the young adults with increasing blood pressure, with APOL1 risk alleles being different between high risk and low risk. And there was really no difference among older age groups. And so the hypothesis was that, in fact, if you were a heterozygote and a young adult, that your blood pressure would be slightly higher. And if you're a homozygote for APOL1, so you carried those two risk alleles, that you would have 2.5 to 2.9 millimeter increase in blood pressure. And that this happened at a younger age onset. And so that's relevant in that perhaps it is true that younger adults are at risk of having chronic kidney disease after they've had decades of hypertension. And this is something that we need to try and understand better because it could have huge implications worldwide. Because of time, I can't sort of go over all the potential environmental factors, but I am going to give you one, just because it's an interesting point that we need to think about. And that's potentially exposed to your pollution. And this is the same group that took a look at hypertension. And they said, let's take a look at the pollution within the exposure within New York City. And you could see that there was potentially an interaction that may show that those who were exposed to greater pollution and they had the APOL1 risk perhaps were also an increased risk of kidney disease. So this is just exploratory, but it was intriguing and definitely something for us to think about in terms of environmental exposure. I'm now going to move to the work I do in Africa and just go through some really big highlights. I don't really want to spend too long here because there's a lot of data that I can't present because it's currently under review. So what we've learned in Africa is that it's really an important place for us to study. Charles Rotimi at the NHGRI has been, the new director, has been a strong advocate, originally from Nigeria, and trying to make sure that we are trying to study genetics within Africa and understand the signatures of selection, understand the infections such as sleeping sickness, migrations, and admixture. It's also been shown that the burden of kidney disease in Africa is enormous. It's estimated to be about 14%. And there's about 1 billion people that live on the continent. So it's a huge number of people with chronic kidney disease. The issue is that if you compare among African Americans of what the rates are of, the frequencies are of the risk alleles, and then you compare to West Africans or East Africans, you can see that it's quite remarkable and it's quite different. But the problem is, again, it's variable about where you're from within the continent. And interestingly, this was done by Sherry Winkler that basically showed if you took trypanosomal infections and said, where's the epidemic? This is the Nile Delta. And you could see that it's along there. You could see, in fact, they have the lowest rates of APOL1. So I think the trypanosomal infection makes sense from a historical point of view. I think we have to try and understand though what's its current context in understanding the association with infection. And similarly, G1 and G2 vary a little bit across the continent. And what's important to note is that West Africa has a higher incidence and East Africa has a lower incidence, but in fact, they still have some. And I only show this quickly because I want people to understand that there are different migration patterns within Africa, whether you're from the Bantu expansion that happened, whether you're from the Nilotic group in Tanzania, or really it's a farming group, or the Khoisan, there's actually differences that happen in your ancestral populations. And because of that, there's a huge variability in the APOL1, all within the continent of Africa. So it's hard for us to think about Africa being monolithic, and even West Africa. Dr. Adu and Professor Ojo are lead in the HC Africa Kidney Disease Research Network. I've worked with Ojo since I was a fellow, a resident. He was my fellow. And so to the residents, remember that you will form lifetime relationships with people that you work with. We've been working in this area now for almost 10 years. We've actually recruited 70,000 participants overall on the continent. We've recruited over 8,000 participants in the kidney cohort, predominantly from Ghana and Nigeria. We've now expanded to include Cameroon, Tanzania, South Africa. There's also, as part of the H3 collaborative, where there's biospecimen storage in 3 different countries to support regional growth in this kind of work. Community engagement is enormous. Paulina Tindana leads it for our group, and it's really been working with faith-based and work-based groups, trying to do education in specific languages, a ton of outreach, and it's really been quite a remarkable effort that they've been able to do within the continent, including ethics boards, including government, and including all the patients. And so it's really quite remarkable the work that they've done. In our Kidney Disease Research Network, capacity building is one of the most important things that we do. When I spend time there, it's predominantly acting as a consultant to teach and to mentor and to support. And as much as we can, we want them to be addressing things that are really important to them. And we continue to work on getting as many trained as possible. So I can only give preliminary data because it is under review, but just in our first sample, when we did the odds of kidney disease, what we found was in our first few thousand, that in fact, the odds of kidney disease were about 1.6 times greater, higher risk of kidney disease among those compared to healthy controls. And now what we're working on is, and is under review, is greater than 8,000 participants. So we'll find out, hopefully we'll be able to present that data next time. But because I couldn't present our data, I wanted to present a recent study that just came out a few months ago. And this is an HIV positive population that are currently being treated, about 2,500 participants who had APOL1 genotyping and measures of kidney function. And in fact, the APOL high-risk genotype was quite low at 6.2%. And what was interesting was when you took a look at this panel on the right, among a West African population in Nigeria, you could see, in fact, the two risk allele difference was enormous, went from 2.1 to 14.5%. So even within West Africa, there's a huge amount of difference by ethnicity. And it's really important to note that. And again, the association about 2.6. So we keep on sort of our regression to the mean. I think it's all going to fall between 1.5 and 2 times greater risk. And so any power calculations we do going forward need to be really powered appropriately. And this is really where it falls with adult Nigerians with HIV. So I really feel that this is where our risk is. And we need to think about what that means when we think about trials and how we're going to plan future studies. I'm also doing a project in Zambia among HIV patients. I'm only showing this because we're interested in a marker called SUPAR, and we think there might be differences in SUPAR levels by your degree of kidney function, but also see if there are differences in the interaction between high-risk alleles versus low-risk alleles, and we're trying to study that there. What's interesting in Central Africa and Zambia specifically is that their G2 is higher. It's almost double the incidence compared to other places. So I think there might be some allelic differences we'll be able to test similar to what we were doing when we talked about the mouse models, that maybe we can see if there are actually allelic differences. So the last part I'm going to go over is actually the part that I think is really important to note, because it brings it back to what should we do in Canada. When we take a look at significant predictors, I've really harped about this 1.5 higher odds ratio, but these are the typical factors associated with rapid progression. And the question is, where does APOL1 improve our prediction? In a multi-ethnic group in Ottawa, and I just use it because I thought it was important to show in the Canadian context, when you take a look at these kidney failure risk equations, you're actually able to see, if I take these 4 factors, age, sex, kidney function, and albumin in the urine, am I able to actually predict an outcome? And what's pretty good is that you're able to predict about 80%, which is actually quite good. And what's important is that you're able to predict among those who've got hypertensive disease and glomerular disease like FSGS. And so it's all pretty good, and it's only 4 factors, and we're able to predict quite well. So the question is, what does adding APOL1 do? And there's only been one study so far that took a look at it, and it really only adds a modest amount. And so this is a complicated slide, but if you just look at the bottom, if you had low risk of disease, perhaps APOL1 may actually help differentiate. But the closer you are and the higher the risk you have, it really doesn't make a difference. Georgia Dunstan, who worked with us in our very original study at Hopkins, really kept emphasizing in this large collaborative that we were in called FIND, that people have to stop talking about discovering a gene among African Americans, because she was worried that people would call this a Black gene. And I think my concern is what I've noticed is that over time, people continue in the United States to describe this as a Black gene, which is completely wrong. What we have to remember is that our hypothesis of the West African genome was that, in fact, that accounted for the higher risk. But we have to remember that 12 million Africans were enslaved. And if you follow actually on the slave voyages, this phenomenal map to look at, just overwhelming to be able to see where people were enslaved. And not surprisingly, that's actually where we see the highest frequency of those who have evidence of APOL1 high-risk alleles. And so it's important to recognize that this is a worldwide issue, that this isn't just an African-American issue in the United States or a West African issue. In fact, we really need to think about populations and recognize that this APOL1 risk is really reflective of where the migration patterns were for the slave trade. Again, Africa is not a monolith, and we really need to think about how do we understand where people come from. And so perhaps we should only be assessing genetic risk regardless of where you come from. And most recently, in an editorial, Vanessa Grubbs, who's a nephrologist, who's now left to really start addressing structural racism within medicine, really keeps saying that APOL1 polymorphisms are contributory, but they're not fully explanatory in racial disparities. And this is really important that we don't get this conflation of genetic ancestry and race. And just to show you that, in fact, in kidney disease, what we're concerned about is people who donate a kidney, let's say a deceased donor who was given a kidney, and they may have high risk alleles within this donor. In fact, they have poor graft survival. And so that's concerning because the question is, you might limit those who actually could donate. And in fact, in the States that they were actually talking about taking the word race within the kidney donor profile index and replacing it with genotype, which I think again has these issues that we have to be very concerned about. And these are just two examples of exactly the same individual, a potential donor for a family member. And you can see their risk is actually very similar at 3.42 if you're white and 3.86 if you're African American, but have the high risk alleles. And so we really want to make sure within nephrology we get this right. And we have to really think about this. I give these examples of persons presenting for living donation or evaluation for a family member. And I assume this would be somebody sitting in clinic, and you could even say African Canadian at this point. But what I try to make the point is that, in fact, if you're from Kenya, you could be from the Bantu tribe ancestrally, you could be from a Khoisan tribe, or you could be from a Nilotic tribe. So you could have 0, 1, or 2 risk alleles. But recognize that they identify as Kenyan or East African, and you may not think that they're at risk. Or they identify as Black, and therefore you think everybody is at risk if they're Black. But that's not true, because those who are Brazilian but may self-identify as white could also be high-risk alleles. So it's really important for us to think about this and about how we address this. What about testing in Canada? Well, I think the key thing is I'm not sure we're there yet. People keep asking me. I haven't brought it. I've been a little bit more conservative than my American colleagues. And I think we need to think about perhaps addressing how to communicate it and to really think about how to screen appropriately. So how do we approach it? I think I've really talked a lot about making sure that you understand that's an important gene to understand. But there's a lot more work that needs to be done. I'm hoping that the trials are really an area that we can focus on in FSGS. And so that would be an individual benefit, but we really need to still think about the population benefit and population screening before we start moving forward. And I keep telling everyone, I think we need these larger collaborative studies, and especially now in COVID-19 and HIV in the States, really trying to understand what the potential impacts are for innate immune function. I'm just going to remind everyone that we still need to address disparities in chronic kidney disease. And as somebody asked me yesterday, to please make sure I talk about that in Canada, we still need equity data to understand these disparities. So I'm going to end just by thanking all the groups, the people that I work with. It's been a phenomenal adventure. Always exciting to go travel and work with them. They're inspiring. In fact, Peggy Hill that I heard about is quite a formidable person from what I hear. But I am proud to be the first nephrologist to hold the chair and to find out that she actually was a nephrologist. And she was a founding member of the Canadian Society of Nephrology. And I think she always put patients first. And that's the hope I do over the next few years. Thank you.
Thanks, Rulan. I've learned a huge amount from this lecture, including I didn't know that she was a nephrologist. So thank you for that additional information on top of that huge body of work that you've worked on across your career that looks like it's so important from so many different perspectives. Obviously the genetics and the interaction between geography and genetics, and also maybe nephrology moving into the world of biologics and targeted disease for both prevention, so targeted therapies for prevention as well as treating disease. So we do have a few minutes left for questions. Just
yeah, my apologies for going late. I really wanted to make sure I address some of those social issues.
If people would have any burning questions, they can raise their hand. Actually, I see that there's a question in the chat box. Amir wanted to know if the recent upheaval in renal function formulas and race have already led to any widespread changes in what formulas are used, and how do you see this changing?
Thanks. So I think for those race-based equations, we're going to come out from the Canadian Society of Nephrology to talk about endorsing that. And our goal is to work with pathologists and labs across the country to try and advocate for that. The one limitation is that the best equation includes a filtration marker called cystatin C, but it's much more expensive. And so what we're concerned about is the impact if we start saying that everybody should get cystatin C. People might not get tested as often because of the cost. And so that's an issue also. And what we're really concerned about with nephrology is that we've actually had positive trials for the first time with all the flozins and the SGLT2 inhibitors for type 2 diabetes. And so we're really interested in making sure that those patients can get access to drugs. So we just want kidney function to be measured often and repeated, and decisions get made on repeated measures, and you don't make a single decision on sort of one value alone.
So is this messaging resonating with the nephrologists across the country, across Canada as well as the US?
Yeah, I think so. I mean, we're definitely talking about it, and our hope is to get something out as soon as possible to really try and change the conversation, like really get it removed. And it's controversial. It was interesting. Amaka Nanye, who I mentioned at the beginning, who talked about it, she and I spoke over the weekend, and she said, she had a lot of people push back, about it in the States too. But, I think in nephrology, we formed a social justice committee at our American Society of Nephrology group, and at our, membership thinks it's important. And I think we need to keep challenging the dogma, right, as much as we can.
Mm-hmm. Do you think that will get right down into the medical schools? That knowledge translation is always a challenging task.
I hope so. I mean, my hope is really to make sure that we are teaching more about kidney disease overall. And then when we talk about kidney disease, making sure that we address this and try to make sure that we're talking about how do you understand the difference in race, ethnicity, and ancestry, right? I think it should be part of the medical school discussion. I think it's important for all of us too, because I think we all need to change that conversation a little bit as well.
Yeah, a lot of food for thought. And I'm sure that if anybody has questions, Rulan would be happy to answer any questions via email. Just a reminder for people to actually complete the evaluation and register their attendance. I think Christian put in the chat box the link that you can use. And normally when we do this in non-pandemic times, we have celebrations of our speaker. We take them out, for dinner, and really celebrate them in a way that we can't necessarily do during the pandemic. But I will be delivering a gift to you, Rulan.
Oh, thank you.
So thank you for just an amazing FM Hill lecture, and many thanks from us here at Women's and for everybody across the network. So thank you, everybody, and do ask Rulan any questions. Keep her busy with feedback on her talk. Thank you.
Thanks so much. Bye. Bye, Rodrigo.